Tama English time.
Parlar geleceğin attığın adımla ileriye doğru, en doğru bu yerde. Kim istemez dünyayı anlamak ortak bir dille. Sınırımız yok, kendini aç, bilgini paylaş herkese. Çünkü lisan güven katacaktır kişiliğine. Bazen kültür gezisinde, bazen iş için gereklidir. Belki sevdanın bir dili, belki de eğitim tercihidir. Gel ve katıl aramıza sen, kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak. Bir güneş üstümüzde parlar, her yer aydınlık ve renklenir. Bütün dünya birleşince bizi de hayalin gerçekleşir bir gün. Yeter ki iste ve ilk adım öğren İngilizcen ve İngilizce. Kafandaki soruları ana diliyle sana destek olabilir güvenilir öğretmen. Eğitiminin kalitesi Türkiye'ye yakışır ve sermayesi yerli bir kurum olduk. Öğrenerek adım adım İngilizce, eğlenerek hayalleri atıyoruz. Sağlam adımlar güvenerek kendimizi, ülkemizi seviyoruz. Sesimizi duymayan kalmayacak. Bazen kültür gezisinde, bazen iş için gereklidir. Belki sevdanın bir dili, belki de eğitim tercihidir. Gel ve katıl aramıza sen, kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak.
Tama English time. English time.
Parlar geleceğin attığın adımla ileriye doğru en doğru bu yerde kim istemez dünyayı anlamak ortak bir dille. Sınırımız yok kendini aç bilgini paylaş herkesle. Çünkü lisan güven katacaktır kişiliğine. Bazen kültür gezisinde bazen iş için gereklidir. Belki sevdanın bir dili belki de eğitim tercihidir. Gel ve katıl aramıza sen kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak. Bir güneş üstümüzde parlar her yer aydınlık ve renklenir. Bütün dünya birleşince bizi de hayalin gerçekleşir bir gün. Yeter ki iste ve ilk adım öğren İngilizcen ve İngilizce. Kafandaki soruları ana diliyle sana destek olabilir güvenilir öğretmen. Eğitiminin kalitesi Türkiye'ye yakışır ve sermayesi yerli bir kurum olduk. Öğrenerek adım adım İngilizce eğlenerek hayalleri atıyoruz. Sağlam adımlar güvenerek kendimizi ülkemizi seviyoruz. Sesimizi duymayan kalmayacak. Bazen kültür gezisinde bazen iş için gereklidir. Belki sevdanın bir dili belki de eğitim tercihidir. Gel ve katıl aramıza sen kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak.
English time.
Herkese tekrardan merhaba. V.Gençomü Öğrenci Kongresi'nin ikinci gününe hepiniz hoş geldiniz. Güzel dönütleriniz için çok teşekkürler. Umarız bugün de ilk gün kadar keyif alırsınız. Bugün ilk konuşmacısını davet etmeden önce ufak bir hatırlatma yapmak istiyorum. Her oturum için katılımcı formunu doldurmanız gerekmekte. Sunum sırasında arkadaşlarımız sohbet kısmında formu sabitleyecekler. Unutmayınız ki katılımcı belgesi alabilmeniz ve düzenlediğimiz çekilişlere katılma hakkı kazanabilmeniz için 9 oturumdan 8'ine katılmanız gerekmekte. Ayrıca hocalarımıza sormak istediğiniz soruları sunum sırasında sohbet kısmına yazabilirsiniz. Bugünün ilk konuşmacısı Yale Üniversitesi'nden Profesör Doktor Ellen Hoffman. Kendisi genetik hastalıklar üzerine odaklanmaktadır. Bugün bizlere Functional Analysis of Autism Risk Genes isimli sunumunu yapacaktır.
I'm sorry, my microphone was off. Today's first speaker is Assistant Professor Ellen Hoffman from the Yale University. The main focus of her research is genetic disorders. Today she is going to present about functional analysis of autism risk genes. Professor, you may begin. Hello and welcome.
Hello, thank you. Thank you for the introduction. Can you hear me?
Yes, yes.
Okay, I'm going to go ahead and share my screen.
Okay.
Let me make sure that you can see my slides. Okay, can you see my slides?
Yes, we can.
Okay, great. Okay, so then I'll get started. Thank you.
Okay.
Thank you for the invitation to talk today and to discuss the work that my lab is doing on the functional analysis of autism risk genes. So my lab is mainly interested in understanding basic biological mechanisms in autism. And specifically, the central questions that we aim to answer are: how does the disruption of what is becoming a growing number of autism risk genes lead to the dysregulation of specific cell types and cell circuits, predisposing to a complex behavioral disorder, which is autism? And second, how can we leverage this growing list of risk genes to identify potential new pharmacological candidates? And what I'm going to tell you about today is how my lab is using this animal, the zebrafish, to begin to answer some of these questions. So to begin, I'd like to start by telling you a little bit about the background of what is autism spectrum disorder. I'm sure many of you are familiar with autism or ASD from sort of the popular news media. It's something that's been talked about often, but not many people are familiar with the fact that autism was one of the first people to describe autism was a child psychiatrist named Leo Kanner in a seminal paper in 1943 called Autistic Disturbances of Affective Contact. And he described a group of boys who had these characteristics. They were happiest when left alone. They didn't pay attention to people, disregarded people, and instantly went for objects. They had actions that were very repetitive, such as shaking their head or focusing on spinning objects. And he came to this somewhat forward-thinking conclusion that these were children who came into the world with an innate inability to form the usual biologically provided affective contact. And that by studying the biology of what was happening to these individuals, this could help us to understand the constitutional components of emotional reactivity. So interestingly, Connor, when he first described or coined this term autism, or applied it to this group of children, he was already thinking that there might be some underlying genetic etiology. And I'll just show a series of YouTube videos that highlight some of the clinical features of autism. So children with autism are often described as being in their own worlds. So here's a child who's focusing on an object and not really paying attention to a group of children off to the side. Sometimes kids with autism...
I'm sorry for the interruption.
Sure.
Are you changing the slides right now? Because we can't
Yes.
see. Okay, because
Oh, I'm sorry. Thank you for letting me know. You can't see any of the slides changing?
now it is changing.
Okay.
I think when you make it full screen, it doesn't change. So I think you just
Okay, so stay this way.
Yes, yes, we can
Got
see
it.
the slides. It's okay right now.
Okay. Were you able to see this slide or no?
No.
Okay, you can see the slides now?
Now, yes.
Okay, sorry about that. I didn't realize that. Thank you for letting me know.
No problem.
Okay, all right, so I'm going to play this video. I'll keep them in this mode then. Kids with autism often are described as being in their own worlds. Here's a child who's not paying attention to a group of kids over here. He's paying attention to an object that he's holding. Kids with autism can also sometimes have soft neurological signs, such as toe walking or low muscle tone. Here's a child engaging in sort of repetitive behavior, moving his head back and forth. Also, which is important for something we'll talk about later, kids with autism often have sensory hypo or hypersensitivities. So they can be very fixated on the textures of materials, very sensitive to light or sound. And here's a child who's focusing on a spinning object, which is very commonly seen in kids with autism. And this is a behavior that could be seen in typically developing children as well, but usually kids with autism will engage in these repetitive behaviors for many hours during the day. And here's a child who's also engaging in some toe walking. He's focused on this object. His mother's talking to him in the background. He also shows some hand flapping, but he's really focused on the object, on the spoon. And here, I don't think you can hear the audio, but the father is sort of calling the boy's name, and he's really not responding. So a lot of times kids with autism can present for hearing evaluations because parents think that they have a problem with the hearing, but it's really a selective attention, and children with autism are not attending to social stimuli. So a few other features of autism can be highly clinically heterogeneous. There's a saying in the clinic, if you've seen one child with autism, you've seen one child with autism. Associated symptoms can include seizures, stomach upset, sleep abnormalities, and importantly, there are no pharmacological treatments for the core symptoms of autism. And so usually in the clinic, when a child with autism would present, usually we're giving treatments for associated symptoms, inattention, hyperactivity, anxiety, aggression, and then the main sort of gold standard for treatment for autism is really applied behavioral analysis, which is an intensive, time-intensive form of behavioral therapy. And the prevalence of autism is increasing. It's now estimated to be 1 in 44 children in the US. Only a few years ago, we thought of it as 1 in 54 children. And there is a predominant higher male to female ratio, which is not well understood. And so at this point, I wonder if I can show the slides. Would you be able to see these animations? Can you see the slides advancing now?
No.
No, okay. All right, so we'll have to go without animations. So one way that we can begin to understand more about the biology of autism is to begin to study the genetics of autism. And so what I'm going to do now is give an overview of sort of where the field started and how far we've come in the last 20 years or so. So I'm going to show a timeline. So about 15, 20 years ago, we only knew of a handful of genes that were associated with autism. And these were genes that were associated with rare Mendelian syndromes, such as FMR1 or tuberous sclerosis. As we developed more advanced technologies, which enabled us to get even more detailed views of changes in chromosome structure through molecular cytogenetics approaches and family-based approaches, this led to the identification of synaptic cell adhesion molecules such as neurexins and neuroligands or shank proteins as autism-associated genes. In 2007, there's a seminal paper by Jonathan Sabat where he found that individuals with autism have an increase in copy number variants or increase in de novo copy number variants. And in particular, these occur at specific regions of the genome, such as 16p11, 15q11 to q13. And then if I'm not able to show the animations, but if we fast forward, one second, I'm going to try to share in a different way. Just give me one moment. I'll be right back. I'm going to see if I can fix this so that you can see the animations. Okay, my apologies. I tried to upload the slides directly, but the file size is too big, so we're going to have to go without the animations. So I'm going to do this. Can everyone see the slides now?
Yes, yes.
Oh, okay. Okay. So what I told you was that in 2007, we then knew about copy number variants that are associated with autism. But then, more recently in 2012, with the introduction of whole exome sequencing, we were able to identify individual genes that were associated with autism, which we began to call high confidence autism risk genes. And these were identified by looking at simplex affected families, meaning there's one affected individual with autism, an unaffected sibling, and unaffected parents. And what these studies found was that there was an enrichment of de novo damaging mutations in the affected child that were not present in the unaffected sibling. And that these were, there was a significant enrichment of these damaging de novo variants in individuals with autism compared to unaffected individuals. And if we could begin to identify which genes were affected in unrelated individuals, this could then lead us to begin to identify a growing list of autism risk genes. And this then led to what is now 102 autism risk genes. So the most recent whole exome sequencing study in autism has now identified over 100 genes that we know are strongly associated with autism risk. And these include genes that have a variety of different functions, including CHD8, which is a chromatin modifier, and SCN2A, which encodes a type 2 voltage-dependent sodium channel. So where the field has come over the last 20 years is from only being able to identify genes that were associated with rare Mendelian syndromes to being able to identify over 100 genes that we know are strongly associated with risk. However, the challenge that we face is now how do we begin to understand what is the underlying biology that links or unites these genes? And one way that we can begin to do this is through co-expression network analysis. And so this was a study that looked at when and where many of these autism risk genes are expressed in the developing brain. And what this study did was they built spatiotemporal co-expression networks around autism, high confidence autism risk genes, and found using the BrainSpan database that these genes tend to cluster and show strong enrichment in mid-fetal glutamatergic projection neurons, identifying a potential point of convergence. In addition, the recent whole exome sequencing study also found that there's an enrichment of expression of autism risk genes in genes from not only the excitatory neuron lineage or glutamatergic neurons, but also inhibitory interneurons. And so we've now come to the point where we're beginning to identify common pathways that might link these different risk genes. However, we're still faced with one of the challenges, which is how do we advance from this list of risk genes to identifying potential therapeutics. And one of the challenges that we have is that each of these autism risk genes gives rise to a range of different proteins or different isoforms, which can affect multiple different cell types and different circuits and predispose to a range of different behaviors. And so what we aim to do is identify particular points of convergence across different risk genes that might represent a point of entry for developing pharmacological targets. And to do this, we've identified zebrafish as an optimal genetic system that would enable us to do this. So our goal in using zebrafish is not to recapitulate the clinical features of autism in fish as an animal model, but rather to leverage key advantages of zebrafish that allow us to do this, that allow us to advance from understanding how a perturbation in a particular gene might affect specific cell types and circuits and predispose to altered behaviors. So there are really three key advantages that we rely on. And one of the reasons that my lab studies them. First, zebrafish are very easy to manipulate genetically. So it's possible using CRISPR technology to inject into a one-cell stage embryo CRISPR's targeting multiple different risk genes and to develop relatively rapidly and at low cost mutants in different genes. Second, zebrafish develop externally, and they have a rapid development. So what you're looking at is a time-lapse video of the first 23 hours of development, and that occurs in a dish. Zebrafish have fully transparent embryos, so it's possible to visualize basic neurodevelopmental processes in real time in a whole brain. And there's strong conservation of neurotransmitter pathways and brain regions. So zebrafish have the same subdivisions of brain regions that mammals have: forebrain, midbrain, hindbrain. And finally, zebrafish are an optimal system to perform large-scale behavior-based screens. And so what you're seeing here is a 96-well plate. We can pipette individual larvae into the wells of a 96-well plate, and then we can track different aspects of their locomotor activity. So we can look at responses to sort of visual startle or acoustic startle, get a readout of that behavior, and ask how that changes in response to exposure to different drugs. And so in my lab, we've now developed a pipeline where we can begin to look at multiple autism risk genes simultaneously using zebrafish as a model. And so what we do is we use CRISPR-Cas9 to target multiple different risk genes. Capitalizing on the transparency of the zebrafish brain, we can then visualize basic processes in brain development, and then we can perform large-scale behavior-based screens to identify drugs that might rescue or reverse particular abnormal behaviors and identify drug candidates. And so we did this in our published work for one particular autism risk gene, which is contagion-associated protein 2. It's a cell adhesion molecule that's involved in neuron-glial interactions and also in the clustering of potassium channels and myelinated axons. What we did was we generated zebrafish mutants of contactin AP2, and we found that these mutants display deficits in forebrain GABAergic neurons. And by tracking their locomotor activity, we were able to identify a specific phenotype of nighttime hyperactivity that occurs in these fish. And using a method called pharmacobehavioral profiling, fortunately, it's not completely displaying clearly here, but we could take the behavioral responses of wild-type fish to over 500 different drugs and ask how the mutant behavioral fingerprint compares and identify potential suppressors of the behavioral phenotype. And when we did that, we found that drugs that had estrogenic activity, surprisingly, such as a plant-derived estrogen biochanin A, was able to selectively suppress nighttime hyperactivity in these mutants, suggesting a new pathway that was not previously associated with this gene. And so now what we've been doing is generating mutants in multiple high-confidence autism risk genes, many of the genes that I showed previously, with the idea that we can then study multiple different sensory processing circuits, and how these genes disrupt these circuits. And in particular, we focused on one particular sensory processing circuit, which is visual startle response and arousal behavior. And so this is work that was primarily has been spearheaded in my lab by a very talented postdoc, Helen Weinschutz-Mendes. We've developed an assay where we pipette zebrafish larvae into the walls of a 96-well plate and then ask how they respond to visual startle stimuli, to dark flash or light flash. And we also test their overall levels of arousal. And this is relevant to understanding how these genes affect sensory processing circuits. And so to give an example of what we're looking at, we acclimate zebrafish to a lights on condition and then turn the lights off in succession Zebrafish have a very characteristic response to lights off where they undergo an O-bend, where their head touches their tail and they swim in the opposite direction. We can do this in high throughput. And so this is an example of our plate where the fish are acclimated to lights on condition and then here undergo response to lights off. And then we can then track the activity of each fish in response to that behavior. And so we can then get a readout of how the behavior of each fish changes following successive dark flashes. We can then reverse the assay and ask how the fish acclimate to a lights off condition and then successive light flashes. We also can track the activity of our fish on a rest-wake cycle and see how their activity is during the day versus during the night. And we can do this across 10 different autism risk genes simultaneously to see how disrupting these genes affects these basic sensory processing behaviors. And what we found is that we're able to cluster genes correctly based on behavioral profiling. I wish you could see the animations in this slide. I'm going to have to try to do this. But what we're doing here is we're showing different behavioral parameters on the x-axis and different mutants on the y-axis. And what you can begin to appreciate is that we generated 2 different autism risk gene mutants for each gene, 2 different independent lines. And each of these 2 lines tends to cluster together, meaning that they have similar behavioral effects. So for example, one of the mutants that affects SCN1LAB, which is the zebrafish version of the sodium channel genes SCN1A and 2A, that these have a very striking behavioral phenotype of daytime hypoactivity and nighttime hyperactivity, as well as an increased response to a lights-on stimulus. In contrast, mutants of another autism risk gene, DYRK1A, show a different phenotype where they have decreased activity during the day, as well as an increased response to visual startle activity. And so now what we're doing is we're leveraging these behavioral phenotypes to begin to identify drug candidates. And what we do is we expose wild-type fish, or normal fish, to a library of over 500 US FDA-approved compounds, and then we expose them to our behavior assays. And what we find is that we're able to correctly cluster drugs based on their pharmacological mechanisms. And then we can map our mutants onto the behavioral profiles of these drugs, which might begin to give us a sense of which drugs would have the ability to rescue or reverse the abnormal behavioral phenotypes in these mutants. And so in conclusion, what we were able to show is that for one of our mutants, estrogenic compounds could suppress the behavioral phenotype and concatenate B2 mutants. We can use this high throughput screening approach to identify sensory processing phenotypes. Pharmacological screens can begin to help us to identify novel compounds linking genes to neural circuits. And as a next step, we can begin to identify suppressors of the behavioral phenotypes and begin to utilize the fish model to do circuit imaging. And as a preview, my lab has been doing work using whole brain activity mapping, which means that we can image the entire brain of a larval zebrafish. This is the telencephalon, tectum, and cerebellum. We can label them for phospho-ERK, which labels all active neurons, or total ERK, which is all neurons. And using a computational pipeline, we can actually map these images onto a zebrafish reference brain, or Z-brain. And this can give us a readout for where there are regional deficits in brain activity in our different autism risk gene mutants. And what we hope to do is link the behavioral phenotypes to the brain activity phenotypes and the drug phenotypes as a way of understanding what links all of these different autism risk genes at a more fundamental level to try to uncover basic mechanisms that are relevant to autism. And with that, I'd like to thank the members of my lab who contributed to this work and our funding support. And I'm happy to take any questions that you have.
Thank you for your presentation. Now let's see if they have any questions. They will appear on the screen. Okay, we have one question. It's from Beril Gürel. Will be autism treated with gene therapies? Is there any study for that?
That's a great question. So there are a number of labs that are looking at whether, so my lab is interested in understanding, how do we understand basic mechanisms that link the different genes, with the idea that maybe there's a pharmacological treatment that would be relevant across different types of autisms. But at the same time, other labs are looking at, just this question, which is, if we can identify what the genetic perturbation is, can we just directly change that? So we don't have the ability to do that yet, but I think there's a line of research that's looking at trying to directly alter, sort of correct the genetic abnormality. At the same time, many of these mutations are haploinsufficient, which means there's one good copy, one bad copy. And so some other work is looking into whether you can actually increase the expression of the good copy to compensate for the mutated copy. And so this is an active line of research. It's not something that my lab has been looking at in particular, but this is a growing area of research.
Another question, Gizem Yıldırım. Thank you for your time. If we understand all gene mutations, will parental diagnosis and treatment be possible?
That's another great question. I think right now, the genes that I was discussing, these sort of high confidence risk genes, account for about maybe 20 to 30% of all cases of autism. So there's still a large percentage that is sort of unaccounted for. So I think that we still have a lot of work to do in terms of understanding more about how common variants might contribute to autism. We probably do know a lot about the rare variants. Prenatal diagnosis is tricky because in some of these cases, usually they're identified sort of after the fact, meaning the child has autism, would undergo whole exome sequencing, and a de novo mutation would be identified. I don't think it's at the level now where we're screening for these known autism risk genes prenatally, but that may be something that would occur in the future as we have a better understanding of which mutations are most likely to contribute. I think now it's still, I think that's still not something that's entered the clinic.
Also, we have some thank you messages from our listeners.
Sure. Well, if anyone has any questions, please feel free to reach out to me on email.
So we have still questions, I guess.
Sure.
It's from Abdullah Özdemir. Is the increase in brain plasticity in autism, especially in Asperger's syndrome, a shield against Alzheimer's disease?
That's a great question. I don't know of studies that have directly looked at that, and probably because we're dealing with different ends of the sort of the age spectrum. So usually in autism, we're looking at very young children. Obviously, individuals with autism become adults, but a lot of the work has focused on very young children. But I don't know of a study that specifically looked at whether individuals with autism are sort of protected against Alzheimer's disease. I think often we think of them as somewhat distinct disorders. But that's a great question. I don't know if someone's really addressed that directly. Although I guess I will say that there is some emerging literature that suggests that sometimes some of the mechanisms that are involved in neurodevelopmental and neurodegenerative disorders may actually have some overlap more than we appreciate.
I think there is no other question.
Okay, sure. If anyone has questions, they can reach out to me. I'm sorry we had trouble with the slides showing,
No problem.
but anyway, please feel free to email me with questions.
Still, we are thankful for your presentation. So
Oh.
thank you for sharing your time with us.
Sure. Thank you. Thank you. Have a good day.
Thank you.
Okay.
Bye.
Bye.
Have a nice day. Şimdi kısa bir arada saat 11'de başlayacağız.
Tama English time.
Parlar geleceğin attığın adımla ileriye doğru en doğru bu yerde kim istemez dünyayı anlamak ortak bir dille. Sınırımız yok kendini aç bilgini paylaş herkese. Çünkü lisan güven katacaktır kişiliğine. Bazen kültür gezisinde bazen iş için gereklidir. Belki sevdanın bir dili belki de eğitim tercihidir. Gel ve katıl aramıza sen kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak. Bir güneş üstümüzde parlar her yer aydınlık ve renklenir. Bütün dünya birleşince bizi de hayalin gerçekleşir bir gün. Yeter ki iste ve ilk adım öğren İngilizcen ve İngilizce. Kafandaki soruları ana diliyle sana destek olabilir güvenilir öğretmen. Eğitiminin kalitesi Türkiye'ye yakışır ve sermayesiyerli bir kurum olduk. Öğrenerek adım adım İngilizce eğlenerek hayalleri atıyoruz. Sağlam adımlar güvenerek kendimizi ülkemizi seviyoruz. Sesimizi duymayan kalmayacak. Bazen kültür gezisinde bazen iş için gereklidir. Belki sevdanın bir dili belki de eğitim tercihidir. Gel ve katıl aramıza sen kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak.
Tama English time. English time.
Parlar geleceğin attığın adımla ileriye doğru en doğru bu yerde kim istemez dünyayı anlamak ortak bir dille. Sınırımız yok kendini aç bilgini paylaş herkesle. Çünkü lisan güven katacaktır kişiliğine. Bazen kültür gezisinde bazen iş için gereklidir. Belki sevdanın bir dili belki de eğitim tercihidir. Gel ve katıl aramıza sen kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak. Bir güneş üstümüzde parlar her yer aydınlık ve renklenir. Bütün dünya birleşince bizi de hayalin gerçekleşir bir gün. Yeter ki iste ve ilk adım öğren İngilizcen ve İngilizce. Kafandaki soruları ana diliyle sana destek olabilir güvenilir öğretmen. Eğitiminin kalitesi Türkiye'ye yakışır ve sermayesi yerli bir kurum olduk. Öğrenerek adım adım İngilizce eğlenerek hayalleri atıyoruz. Sağlam adımlar güvenerek kendimizi ülkemizi seviyoruz. Sesimizi duymayan kalmayacak. Bazen kültür gezisinde bazen iş için gereklidir. Belki sevdanın bir dili belki de eğitim tercihidir. Gel ve katıl aramıza sen kırılacak tüm ön yargıların. Birlikte başaracağız engelleri aşarak.
English time.
Herkese tekrardan merhaba. Sıradaki konuşmacımız Çanakkale 18 Mart Üniversitesi'nden Sayın Doktor Öğretim Üyesi Çağrı Ünal. Son zamanlarda deneysel psikoloji üzerinde çalışmalarını sürdüren Çağrı Ünal, kortikal ketleyici şebekelerin evrenselliği üzerine lateral amigdala bize neler söylüyor adlı sunumunu yapacaktır.
Our next speaker is Dr. Çağrı Ünal from Çanakkale 18 Mart University. The main focus of his research is experimental psychology. Today he is going to present on the universality of cortical inhibitory networks. What is the lateral amygdala telling us? Professor, you may begin. Hocam hoş geldiniz.
Hoş bulduk arkadaşlar. Sizleri iyi görüyorum. Çok mutlu oldum. Öncelikle nazik davetiniz için çok teşekkür ederim. Sunuma başlamadan önce şeyi söylemek isterim. Terminolojide çok sıkıntı yaşıyoruz Türkçe'de şu anda. Hani oturup sözlük çevirisi gibi yapmak da zor oluyor. Bir İngilizce bir Türkçe'de yapınca sunumun akışı bozuluyor. O yüzden sizlerin de affına sığınarak sizi biraz kötü İngilizceme, kırık İngilizceme maruz bırakacağım. İngilizce sunum yapmayı düşünüyorum. Umarım problem olmaz. Dediğim gibi böyle daha sağlıklı olacağını düşündüm. Umarım Türkiye'de bilim yaptıkça daha rahat Türkçeleştirme yapmak mümkün olacak ileriki yıllarda. Ama tekrar teşekkür ederim nazik davetiniz için.
Biz teşekkür ederiz hocam.
O zaman ben ekranımı paylaşıyorum.
Tamam hocam.
Peki hocam.
Tamam. Now, I hope you can all see my screen, and I hope you are not having any trouble seeing my presentation. So, basically, the English translation for the talk is going to be as follows regarding the universality of cortical inhibitor networks, and what does the lateral amygdala tell us about that. First of all, I want to reiterate that I'm very grateful for your invitation and giving me the chance to introduce myself and some of my work to you as part of this effort. So let's start. So first of all, I'd like to give you some outline of my talk today to show you what you are going to encounter. So I'll basically begin with a prelude. So I'm going to go over some basic definitions and pieces of information. Since I don't know your level of neurobiology knowledge, I'll try to give some hints so that it will be easier to follow the rest of the talk. But please do not hesitate to ask questions you may have, no matter how simple or sloppy they may sound. No question is a bad question. So I would be very happy to answer any questions you may have. So once I do this basic definition section, I will just move on and I will describe you in a very coarse manner the cortical areas, what I mean by cortical areas. I'll talk about the types of cortical areas, and I'll talk in particular about the cerebral cortex, which is the most representative form of cortical areas, maybe when it comes to humans, because it's one of the latest inventions of the evolution. And I'll be going over the cell composition of cortical areas. I'll talk about their morphology and electrophysiology a little bit, and it will be followed by a definition of inhibitory interneurons. There, most of those this information comes actually from in vitro studies, in vitro patch clamp studies. So at the end of the talk, maybe I'll be able to answer questions you may have regarding the in vitro patch clamp technique. Then I'll move on with the definition of the amygdala. At first, I'll take a step back and I will talk about the functions of the amygdala. We are all probably familiar with the functions of the amygdala from popular magazines about its involvement in fear learning, etc. So we are going to go a little bit over that. And there is a section of the amygdala, the basolateral amygdala, which is a cortical-like structure in terms of its cellular makeup, but unlike the cortex, it lacks a layered organization. And I was particularly, along with my collaborator Ben Günal and Meglin Bolton, interested in whether this lack of layered organization also means differences in the inhibitory network structure. And let me say the last message first. There seem to be some universal principles. So, first of all, as part of my prelude, I want to define what I mean when I say principal neurons and interneurons. In the most classical sense, from the neuroanatomy that comes from the beginning of the 20th century, principal neurons were basically neurons that were sending very long axons down from the motor cortex to the spinal cord, upper motor neurons, then from the spinal cord to the muscle, lower motor neurons, and the dorsal root ganglion neurons that send back information about the sensory niche the organism exists in. And this is a very broad definition. And with advances in staining techniques, the way we see principal neurons, the way we describe principal neurons and interneurons changed quite markedly. So with advances, we could see subregions within different brain locations. And we call them regions, or we call them nuclei, not to be confused with the nucleus of the cell. So basically, a brain structure, a brain region, is a collection of cells that share some sort of cytoarchitectonic characteristics, some sort of functions, and they share their input and output relations, which is referred to as their hodology. So one problematic aspect of neurobiology is that the functional and anatomical characterizations do not always overlap, but sometimes we have to stop being perfectionists and we have to define brain regions. And when it comes to that, when we talk about the communication between two regions, the communication, the long-term, long-range communication is carried out by the principal neurons. But principal neurons do not explain all the story, because you also need some sort of a local regulation of these principal neurons to preserve information fidelity, because no cell is perfect, and when the information comes, sometimes the principal neuron might over-represent the information. The changes in the principal cell might outlast the input. And to control for that, interneurons developed, evolved, and they control the output of particular regions in a very local manner. And usually the interneurons do not send long-range axons. But of course, there are exceptions. Actually, there are exceptions to everything I'm going to say today in this presentation. This is what science is about, but I just warn you, take the things I say with a grain of salt. That's my disclaimer that I would like to do before I begin further delving into the issues. And there are important exceptions, but interneurons are largely inhibitory, which in other words means they are supporting the cessation of ongoing signals for a variety of different regions. And let me tell you what I mean by excitatory and inhibitory. So the principal neurons are usually excitatory. They release the neurotransmitter glutamate, which I marketed as their receptors with blue. So when glutamate binds to their ionotropic receptors, the receptors change conformation and they become permeable to cations like sodium, potassium, and calcium. And those cations have a reversal potential much above the hyperpolarized membrane potential of the neuron. So basically, glutamate transmission drives the cell closer to the action potential threshold and sometimes beyond it, and makes the cell fire action potentials and relay their signals further. Of course, there are glutamate receptors that are metabotropic. I'm not going to talk about them today. And here above, I hope you see my cursor. We have two traces from recordings. The bottom one is the action potentials induced in the presynaptic neuron. And the presynaptic neuron, as you can see, results in depolarizations in the postsynaptic neuron, which is a function of cation influx into the cell. Interneurons, on the other hand, typically release a neurotransmitter called GABA, which is converted from glutamate through glutamic acid decarboxylase. And GABA, when it binds to its ionotropic receptor, changes the ionotropic receptor's conformation to that chloride ions in. Chloride is negatively charged and usually hyperpolarizes the cell, and the chloride reversal potential is well below the action potential threshold. So GABA transmission reduces the likelihood that the cell is going to fire. And in this example, you see that a GABAergic neuron firing action potentials hyperpolarizes the postsynaptic cell, driving it further away from the action potential threshold and making the cell less likely to fire action potentials. And here you see the concentrations and equilibrium potentials of different ion species. There are a zillion types of different inhibition, so the computational power of the brain is enormous. But I'm just going to simplify the matters a little bit, and I will talk about two types of inhibition, which is feed-forward inhibition, you see on the left panel A, and recurrent and lateral inhibition you see on the panel C. So I hope you can follow my cursor. So this segment represents the axon of a presynaptic neuron that brings information from another brain region and releases glutamate in the local region that we focus on. And the B is the dendrite of the postsynaptic principal neuron. So it gets excitation from this neuron. But the same presynaptic axon terminal arborizes in the brain region that it innervates, and it also targets an interneuron, which in turn targets the postsynaptic neuron that was originally targeted by the same presynaptic axon. In a sense, this type of feed-forward inhibition dampens the information. So imagine you see a visual scene that lasts for like 1 second, and the cell that is going to process this information also has like inherent ion channels that could prolong the signal. But you don't want the signal to be prolonged because you want to represent the outside environment with high fidelity. So that type of feed-forward inhibition actually dampens the input and dampens the internal ability of the cell to start a regenerative process. So in this way, you have fidelity of signals. Then you have the other type of inhibition, which is recurrent or lateral inhibition. And I'm not going to very much delve into details about the differences between recurrent and lateral inhibition, because they are not only anatomically specified things, but they are also state dependent. But in short, the cells in a particular region receive excitatory input from another region, and when those cells send their axon to the other region, they also send another collateral to a local interneuron, which in turn inhibits the cell that originally sends the message and the neighboring cells. So sometimes the neighboring cell is comparatively more inhibited as compared to the cell that receives the original signal. In a sense, it reduces the signal to noise ratio, and people call it winner take it all model. So and it is especially important when it comes to our ability to differentiate between different stimuli. So this is a very general form of inhibition too that exists in cortical and subcortical regions. So let me talk briefly about the cortical areas of the brain. When we say cortex, actually the first thing that comes to our mind is the outer bulk of the brain. So the cerebral cortex is a six-layered neocortex. Not all neocortical regions in the adult animals have 6 layers, but during embryologic development, they necessarily start out with 6 layers. And the cerebral cortex also includes some small portion of allocortex, which is the type of cortex that has less number of layers and is a little bit evolutionarily can be considered as evolutionarily archaic. And I'm going to skip the details about allocortex here, but you might have heard like popular brain regions like the hippocampus, for instance. It's a kind of allocortex that is considered as archicocortex, and is composed of 3 cortical layers. But all those layered structures share some important psychoarchitectonic properties. So let's look at the cerebral cortex. This is the classical image you will see even in high school books. So in the frontal part, you have the frontal lobe that is divided from the parietal lobe with what we call the central sulcus. Then here you have the Sylvian fissure that divides the temporal lobe from the parietal lobe and the frontal lobe. Then in the back, you have the occipital lobe, which contains actually the primary visual cortex around a fissure called the calcarine fissure. So, and we have there the Wernicke's area, the Broca's area, areas responsible for the production of sleep. We have the primary motor cortex that contains the upper motor neurons. We have the primary somatosensory cortex that helps us to sense the world around, the tactile world around us. We have the auditory cortex there to process information, etc. And most of this information to the cortex is relayed through the thalamus, a subcortical telencephalic structure. When we come to cerebral cortex, we can easily see its layered structure. So it has 6 layers. Most cortical areas have 6 layers. And the thickness of 6 layers is also dependent on the function, particular function of that neocortical area. For instance, you see the region A here in the macaque monkey, and you see that the layer 4 is a little bit more elaborate as compared to other regions, the regions B, C, and D, because layer 4 is actually the layer that receives sensory information. And macaque monkeys are also visual animals, and the visual cortex receives a rich array of sensory information, and that's why it has like also subdivisions within its layer 4. So this is very important, and it points out to the labor division among different cortical layers. And when we look at cortical cell types, those cell types are not unique to the neocortex. You are going to see them also in other layered cortical structures, like the hippocampus or the olfactory cortex, the olfactory tubercle, etc. So you have a type of principal neuron that comes in two varieties morphologically. The abundant morphology is the pyramidal neuron, which has a cell body that resembles the shape of pyramids. And there are stellate cells, and stellate cells are particularly important because they are usually found in the layer 4 of the neocortex, and they are specialized for receiving sensory information, relatively unfiltered sensory information from distinct thalamic subregions. Cortical pyramidal neurons also have a unique electrophysiological signature. If you apply a square pulse, square current injection, they are going to start firing. But as you can see, the firing becomes slower and slower over time. That's called spike frequency adaptation. So they are initially going to exhibit a relatively fast firing rate, followed by reductions in the firing frequency. And they constitute about 90 to 95% of principal neurons in the cortex, depending on the species you study. Then, on the other hand, we have the interneurons. They are, in terms of numbers, lower in numbers, but they have like very intricate axonal arborizations, and that's why they can target the principal neurons in a divergent manner. And there are a variety of ways to define interneurons. You can define them based on their cell morphology, somatype, or the number of dendrites they have, or which region in the postsynaptic cell they target. For instance, there are some cells that target the axon initial segment, and they can veto action potentials easily, but there are some cell types that target the dendrites, and they modulate the strength of excitatory postsynaptic potentials coming from other excitatory neurons. They also have very distinct firing patterns. For instance, you have the FS neurons, which are the fast spiking neurons, and they fire at very high frequencies. They can fire up to 50 to 100 action potentials per second. Then you have the late spiking neurons, for instance, and the late spiking neurons have like a conspicuous delay in their spiking onset latency because of a particular overexpression of some sort of a potassium channel that gets activated at critical voltages. Then here I want to mention the low threshold spiking cells. The low threshold spiking cells initially perform some burst of action potentials, followed then by action potential adaptation of frequency. And those cells also have their own unique neurochemical phenotype, and it is quite complicated. You cannot make a one-by-one correspondence, but there are some general rules. For instance, the fast spiking neurons that I just talked about usually belong to the parvalbumin cell category. And those parvalbumin cells are basket cells. They form baskets around the soma of the postsynaptic neuron. And the low threshold spiking neuron I show here usually corresponds to the somatostatin positive neurons. And let me briefly mention, somatostatin is a neuropeptide that is released by the presynaptic terminals, and it can do a variety of functions through metabotropic receptors, which are usually inhibitory in nature as well. Parvalbumin is not something released from the cell. Parvalbumin is a calcium binding protein, and it is possibly very critical in determining the firing pattern of the fast spiking neurons that express parvalbumin. In the cortex, in the neocortex, especially the group by Markram, and there was a guy in Colombia, I forgot his name, they worked very hard on that, and they found, they encountered an interesting phenomena. If you record two principal cortical neurons, if you make the cortical neurons fire at high frequencies, you will sometimes, although rarely, see monosynaptic excitatory postsynaptic potentials because the principal neurons release glutamate. So locally, the principal cells also interact with each other. But what you see more common is this excitation is followed by inhibition. And we know that the principal neurons do not release GABA. So here the picture is the principal neuron both targets another principal neuron, releasing glutamate onto it, exciting it, but at the same time, it releases glutamate onto an interneuron, which in turn targets the other principal neuron and inhibits it. And it took some time to people to discover the neurochemical identity of the cell. Don't let the chart's complexity deceive you. Of course, it's still an oversimplification, and we do it for the time's sake. And we see that the somatostatin expressing neurons play crucial roles in mediating recurrent inhibition in cortical circuits, while parvalbumin expressing fast spiking neurons play a role in feed-forward inhibition. Now I take a step back and I will talk about Pavlovian fear conditioning in rodents. So I'm not going to go over all figures because I see that I'm not a professional in time management. But when we are born, we don't have much inherent fears. And we learn most of the fears by experience. And this is modeled in rats. So when you basically pair a tone that doesn't scare a rat or a mouse or whatever species of interest you want to use, if you pair that tone with a food shock, with sometime the tone comes to elicit also fear responses. So fear is acquired to the tone. And this is thanks to an intact amygdala. So the amygdala is involved in fear conditioning. When we come to the amygdala, it's actually a heterogeneous set of nuclei. So functionally speaking, the lateral amygdala is the station for sensory input. So it receives both sound information and shock information. Sound information is at first not able to excite the lateral amygdala neurons sufficiently, but once it gets paired in a short time window with the food shock, the sound can also start exciting the lateral amygdala neurons. The lateral amygdala neurons then send the signal to the basolateral amygdala nucleus, which is implicated in the reprocessing of sensory input. Then the basolateral nucleus sends the information to the central nucleus, central amygdala, which is the output station of the amygdala that sends information to the lateral hypothalamus perioaqueductal gray type of regions, which are implicated in defensive behaviors like freezing or jumping or escaping. And the central amygdala is another story. It is, in terms of its cytoarchitectonics, it's the striatum-like structure. Striatum is the structure that Bengi talked about yesterday, Bengi Hoca talked about yesterday. But the basolateral portion of the amygdala resembles the cortex a lot. So lateral amygdala being the sensory recipient portion of the basolateral amygdaloid complex contains stellate cells that are very much like the layer 4 neurons in the neocortical regions. Here I show a layer 4 recording and filling, and you can see a stellate form cell, the multipolar cell. And in layer 4 neurons, it is not uncommon to encounter that type of interneurons. And as you can see, like the principal neurons of the neocortex, they exhibit some spike frequency adaptation. And once the plasticity in the lateral amygdala takes place as a result of recording, the tone comes to elicit, as I said, strong spiking in the lateral amygdala neurons. So before fear conditioning, during the presence of the tone, we see scarce action potentials, but after fear conditioning, the number of action potentials rise dramatically. So the tone is able to elicit a fear response. And believe me, the same thing is happening in us. So when we recorded the monosynaptic excitatory connections among LA principal neurons, we found that only 2% of the neurons we recorded are monosynaptically connected. So firing the presynaptic principal neuron in the lateral amygdala results in excitatory postsynaptic response barrages, which is followed by inhibition. And in most cases, there's not even a monosynaptic connection, but we see an indirect inhibition. We know that this is not a monosynaptic response because the response onset latencies are very variable, and the inhibitory postsynaptic potential is sensitive to glutamate receptor blockade, which means that the presynaptic cell actually activates an interneuron through glutamate receptors. And it was pretty common to see that in 15% of the cases, we see a polysynaptic connection among LA principal neurons. So they inhibit each other indirectly. And sometimes we, in our recordings, encountered reciprocal connections between interneurons and principal neurons. You see here two patch clamp pipettes. So the principal neuron excites the interneuron, and the interneuron in turn inhibits the principal neuron. And as you can see, you need a very high frequency activity to get the EPSPs bigger, which means that actually it serves as a high pass filter, which is also the case in other neocortical regions. So to discover the neurochemical identity, we used transgenic mouse strategy. Basically, we used TD tomato animals. So it's a Cree reporter tool strain that has a LOX-P flanked stop cassette, preventing the transcription of TD tomato, which is a red fluorescent protein. And when we made those animals with Cree-specific lines, expressing the Cree enzyme under the promoter of somatostatin or parvalbumin, we can make the somatostatin and parvalbumin cells selectively red. And here we have a staining. So from a somatostatin Cree TD tomato animal. So the red cells are the somatostatin cells in the lateral amygdala, and the green cells is the immunocytochemical signal for parvalbumin immunocytochemistry. So basically we see distinct populations. And then we made paired recordings between somatostatin positive interneurons with principal cells. We found mutual connectivity in most of the cases, suggesting strongly that the somatostatin neurons are implicated in this type of recurrent inhibition. And you see a reconstruction here. This is the somatostatin cell, which has a isoform soma, which is also referred as the Martinotti cell, named after its discoverer. And we can clearly see that the inhibitory postsynaptic, the putative inhibitory postsynaptic contact points are on the dendrites, which matches the scenario in the neocortical regions. And as a last experiment, we did a very crude but smart experiment. What we did was we stimulated the BLA, the portion that is recipient of LA neurons. So by doing that, we could antidromically induce spikes in lateral amygdala principal neurons, and you can see this antidromic spikes. And here you see the principal neurons physiology. And when a spontaneous spike occurred just before the stimulation, the antidromic spike was not seen because of the inactivation of sodium channels, which is a proof that the spikes we saw were antidromically elicited spikes. And when we antidromically activated the lateral amygdala, we found that many somatostatin positive cells responded with an action potential to the internal activation of the lateral amygdala circuitry, whereas we saw that less often in parvalbumin positive cells. And sometimes we also had negative results, but this is because somatostatin positive and parvalbumin positive neurons do not represent a single category. And when we compare their electrophysiological signature, actually we could see striking similarities between cortical interneurons and the interneurons of the lateral amygdala. Here, for instance, you see a parvalbumin cell we recorded, and you see very high frequency spike discharge around 50 to 100 hertz, depending on the level of current you inject. And you can see that the spikes are showing some reduction in their amplitude. And in the cortex, a very similar amplitude reduction is seen with depolarizing shift in the after hyperpolarization amplitude. And you see here a very quick voltage relaxation when the cell is injected with a negative current. On the other hand, somatostatin cells exhibit an initial high discharge of spikes because of specific calcium channels they have, followed by spike frequency adaptation in the lateral amygdala. And the same thing holds true for cortical somatostatin cells. And we see this voltage relaxation, which is more gradual as compared to the parvalbumin cells. And that gradual nature is also preserved in cortical regions. So in conclusion, basolateral amygdala is similar to its layered counterparts when it comes to neurochemical, morphological, and electrophysiological makeup of its constituent neurons that we knew already, but our studies basically have shown that there might be also similarities in the connectivity patterns, which is very important because those cells are also implicated in neurodevelopmental disorders that involve emotional disturbances. And probably somatostatin neurons are going to be important targets for emotional disturbance bearing diseases. And I'd like to acknowledge Bengü Ünal, my collaborator on the project, and Dr. Martha McLean-Bolton, who was the PI of the project. And I'd like to thank you for your patience for during my talk, and I hope I could make some sort of sense. And in the remaining time, I can take your questions if you may have any. And you can ask the questions also in Turkish.
Bu güzel sunumunuz için çok teşekkür ederiz hocam. Arkadaşlarımızın sizin için sormuş olduğu sorular bulunmakta. Bunları ekrana yansıtacağız. Aynı zamanda sizin için de okuyacağız soruları.
Tabii.
İlk sorumuz Sena Özçelik arkadaşımızdan. Cinsiyet hormonlarının amigdala üzerinde etkisi var mıdır? Eğer varsa bu hormonların az veya çok salgılanması amigdalayı nasıl etkiler?
Cinsiyet hormonları ile ilgili reseptörler genelde amigdalada bulunmuyor ama daha çok diğer bir kortikal bölge dediğimiz hipokampüste çok bulunuyor ve hipokampüsle amigdala arasında aslında karşılıklı bir ilişki var ve korkuların genellenmesinde veya tam tersine korkuların segrege hale getirilmesinde amigdala ile hipokampüs arasındaki ilişki çok önemli. Bu bağlamda bu cinsiyet hormonları hipokampüs üzerinden amigdala üzerinde belli etkiler yapma potansiyeli barındırıyor. Öğrenmede, korku öğrenmesinde cinsiyet farkları da çok görülebiliyor bu arada. Dişi hayvanların östrüs cycle'larına göre farklar görülebiliyor.
Diğer bir sorumuz Beril Gürel arkadaşımızdan. Sunum için çok teşekkürler. Sinir hücrelerinin moleküler evrimi incelenerek psikolojik ve mental rahatsızlıklara yeni çözümler bulunabilir mi? Bu şekilde kesin çözüme ulaşan örnekler var mı?
Bu şekilde kesin çözüme ulaşan örnekler yok. Ama özellikle internöronlarda evrimsel olarak farklılıklar görülüyor. Yani primatlara geldiğimizde internöronlardaki RNA ekspresyon patternları tamamen değişiyor. Daha fazla komplikasyon barındırıyor. O yüzden aslında belli başlı hastalıkların modellenmesinde aslında fare veya sıçanların ne kadar etkin olabileceği de bir soru konusu. O konuda daha yüksek hayvanların incelenmesinde fayda olduğunu düşünüyorum.
Teşekkür ederiz hocam. Sanırım başka sorumuz yok. Şimdi arkadaşlarımızın teşekkürler yorumlarını da ekranda görebilirsiniz.
Çok teşekkürler arkadaşlar. Pazar günü uyku saatinde beni dinlediğiniz için.
Hocam bir sorumuz daha var galiba.
Tabii.
Fobi ile
Fobi
amigdala arasında nasıl bir ilişki var?
Yani Nasrullah, sorun için teşekkür ederim. Doğduğumuzda gerçekten çok az şeyden korkuyoruz. Yani bizi yeni doğan bir bebeği korkutan şeyler aslında yüksek ses ve 3 boyutlu görmesi geliştiği anda yüksek yerler. Onun dışındaki bütün fobilerimizi aslında başkalarının deneyimlerini gözlemleyerek veya kendi deneyimlerimiz çerçevesinde öğreniyoruz ve bu öğrenme amigdala sayesinde oluyor. Amigdalası kalsifiye olan bir kadıncağız vardı. Yani çok spesifik olarak bir lezyon vardı. Herhangi bir korku öğrenmesi yaşayamıyordu. Yani işte hayvan dükkanına gittiğinde yılanları kolayca elleyebiliyordu. İnsanların tehditvari sinyallerini algılayamıyordu. Ve bu kadının başından birçok evlilik geçiyor ve birçok kez aslında şeye maruz kalıyor. Şiddete de maruz kalıyor korku sinyallerini öğrenemediği için. Bu tabii adaptif bir şey. Tabii fobilerin hepsi adaptif değil. Bazen e, o segregasyon, amigdaladaki inhibisyondan kaynaklanan segregasyon tam iyi olmadığı için bazı korkuları gereğinden fazla genelleyebiliyoruz. O yüzden de fobiler geliştirebiliyoruz. Ben teşekkür ederim.
Teşekkür ederiz hocam tekrardan.
Başka bir sorumuz yok galiba hocam, bitti.
Tabii. Çok teşekkürler. Prompter'a ihtiyaç kalmadı. Güzel.
Sunum için teşekkürler hocam.
Ben teşekkür ederim arkadaşlar. Görüşmek üzere ileriki
Görüşürüz
şeylerde.
hocam.
Teşekkürler hocam. İyi günler.
Görüşürüz. Kendinize iyi bakın. İyi günler.
Sunum için hocamıza tekrardan çok teşekkür ederiz. Birinci oturumumuz sona erdi. Akşam saat 18.30'da çekilişlerimiz ile ikinci oturumumuza tekrardan başlayacağız. Ödülleri kazanabilmek için çekiliş esnası bekmektedir. Saat 18.30'da tekrardan görüşmek üzere.